Good morning. I'm very happy to be here. My name is Leo Saul. I'm the co-founder and CEO of Saga Diagnostics. We are a three-year-old startup company, a spin-out from Lund University. We are based in southern Sweden, and we focus on cancer diagnostics and a very exciting area called the cancer liquid biopsy. So we all are well aware that cancer is a huge problem, but I want to draw our attention to a number of issues that our company is addressing. One is that the current standard of tissue biopsy is actually insufficient. It has risk, it can miss the tumor, it can lead to complications. Today, it's also very difficult to measure how a patient's actually responding to therapy. The best imaging and tumor markers today are not sensitive and specific enough. And what this means is that many patients who develop recurrence of the cancer, this is detected much too late, and pretty much across all cancer types, metastatic cancer is incurable. At the same time, there's a well-recognized issue that many patients are actually being overtreated. This is sort of the flip side of the coin, where patients who have low-risk cancer go on to have very my mic went out, or it's back. Very toxic therapies that they don't actually need because they are cured by the surgery and local therapies. And finally, it's well known that drug development is quite expensive, in particular within oncology. So the solution to this, we believe, and I think many in the industry believe, is in large part the liquid biopsy and the circulating tumor DNA biomarker. This is very simple to collect from normal blood samples non-invasively. It's quantitative and gives a readout of is there cancer in the body and how much cancer is in the body, and also which mutations are present, and many new therapies target specific mutations. We and others have shown this to be very prognostic and also predict which therapies will work. And we can detect metastatic disease in some cases up to 3 years before it shows up by standard methods. We think that this can be used in the future to spare patients from toxic therapies if they're already cured, so we give the right drug at the right time. And through a combination of these effects, we believe that this technology can accelerate drug development and help drug developers to conduct clinical trials and learn more from their patients and optimize their future drug development plans. So what is circulating tumor DNA? This cartoon on the left shows tumor cells, and the ctDNA is little bits of DNA which escape from the tumors into the bloodstream and mix with all of normal DNA that everyone here has also in their bloodstream. And because it's a mixture of both tumor DNA and normal DNA, one needs very specialized technologies to detect this. We use the tumor-specific mutations and the rearrangements of the DNA chromosomes as biomarkers for a patient's cancer. So we have 3 innovations that address and detect these biomarkers in different ways, which I will not go into today, but to just jump ahead, these are the highest performing technologies out there. Proprietary and patent pending. We have very good validation data, and essentially all cancers release this ctDNA, so it can be applied to any cancer type. Even Forbes has recognized liquid biopsies as a disruptive technology for healthcare by 2020, and I think that's actually not really that much of an exaggeration. This can be used in many areas of oncology. It's a nice figure from nature. And as a company, we are focusing on the part once a patient has a cancer diagnosis. So how can we profile this cancer better and to know if it's a high-risk or low-risk patient? Can we detect if there's any residual cancer after surgery? How is the patient responding to therapy? And can we detect metastasis and tumor evolution over time? We can do this better and faster, cheaper, and also at scale compared to competitors. And one of the main features is our ultra-sensitivity. And what this really means is that we are about 100-fold more sensitive than competing technologies. And a key figure is this lower limit of detection. That is, what % of the DNA has to be mutated from the cancer for you to detect it. And so we can detect the smallest amounts of DNA from the tumor at a much higher sensitivity than the competition. We can do this very rapidly within just a couple days of receiving a sample, very cost effectively, and also we can make this into kits for wide distribution to labs around the world. This addresses many customer needs. First and foremost, the patient. We can get better diagnostics to them more comfortably so they get the right treatment. Of course, physicians and hospitals want to give the best treatments for their patients. Health insurers want to reduce costs, so you don't want to apply therapies if the patients don't need them, so you can save cost. And pharma companies, of course, want to find more patients that have their drug target. They want to conduct their therapy, their trials more efficiently. The market for this is very large. I apologize that there's something with the slides on this computer, but in Europe we estimate the market to be about 9 billion euros. Piper Jaffray Investment Bank estimated the US market to be about 6 billion euros. So together, this is about a 15 billion market opportunity in just these two markets alone. We are focusing initially on breast cancer and lung cancer, two rather large diagnoses that represent over 2.5 billion euros per year. Our business model has sort of a stepwise progression where we operate a central service lab and actually have early adopters already sending us samples. We also are partnering with other labs to set up our technology in partner labs to offer to their customers and their patients. And through this activity, we get some cash flow. We accelerate our own internal R&D and gain insight into the customer base, which feeds into the long-term development of CE IVD marked kits for sale. So the sales matrix for this, our approaches are very large with our 3 different technologies, which have different benefits in different situations and the many different types of cancer that there are. So this market that we approach initially will, of course, expand as we grow as a company. We have a terrific team. Myself, I have an MD and PhD background from top institutions in the United States, like Columbia University, NIH, and Sloan Kettering. I'm actually a cancer survivor myself, so I have a real dedication and passion to bring our technology to benefit patients. Our co-founder is Anthony George, who helped develop the technology. Åke Nilsson is our BD director. He's here in the audience. Please come see him if you have any questions. And Carolina has many years' experience with clinical diagnostics. We have a terrific board of directors with expertise in startup companies, in taking companies to the stock market, IPOs, entrepreneurs, BD, as well as Henrik Winter, who's a veteran within cancer diagnostics. We also have a very good scientific advisory board. I can mention that Dr. Boris Sandale is the former president of the European Association for Cancer Research, so she's one of the biggest names in cancer research. And Niklas Lohmann has been head of diagnosis of a number of diagnoses for the southern part of Sweden. We're also part of a very interesting and helpful network called Nomen, and have advisors who are very esteemed. Carl Borbeck started many companies, Peter Benson within venture capital, and Alva Kaikkonen, who has founded a number of medical device companies. This is a very hot area. We're not the only player. There are quite a number of them, and there's been a number of acquisitions over the last few years. I won't go through all of these, but several have been acquired by Roche, for example, for over $1 billion US. This is a kind of busy slide, so I won't step through all the details, but we of course, have a good sense of what the competition is and can see that we have a quite unique position in a number of key areas. Our development plan spans over a number of years, and we are doing quite well. I won't step through all of this other than to say that we raised capital last year and will raise more money this year and are already talking to a number of different types of investors. We plan to initiate our own clinical studies towards the end of this year to further establish particular technologies as part of clinical practice. Just to give us some recent highlights, we are quite visible within the field with many presentations, including TV appearances and so forth. So people are very aware of us within pharma and academia. We've won a number of awards. As I mentioned, we raised seed capital last year of about 1 million euros. We've also received a number of grants from the Swedish government. And for example, I've been named a top 7 finalist in a startup competition of over 4,500 entrants. Our IP situation is quite good. We expect our first granted patents to come in the first half of this year. We've filed in 14 jurisdictions around the world. We're expanding. We're hiring more people right now. We have a number of early adopters. I'll mention just a few. We have a Swedish hospital partner that is evaluating our technology for use with patients. And we have a number of rather big name industrial customers. We've been chosen to participate in a phase 2 clinical trial with Wint Research, a Swedish biopharma company. So with that, I'll summarize that we are a young, dynamic company that is really on the right track. We're very motivated to bring our technology to benefit patients. We spun out from successful research at Lund University. We have these superior methods for detecting circulating tumor DNA. We have market traction and a good development plan. We have this 3-part business model with a service lab, lab-in-lab partners, and then kits. The market for our technology is very large. We are raising capital this year. And we foresee that as we execute successfully as we have, we may be an attractive acquisition target. And certainly we are thinking about future raises, whether it be through a Series A or even going on the public markets. So with that, I thank you for your attention, and I'm happy to take any questions.